So please note for your quiz on Monday that we'll be doing the first two sections up to, but not including proofreading and repair of DNA. So basically structure and replication. Anything about lab, Dr. Martin?
Yes, those of you who had lab on Monday, I failed to collect your answer sheets. You can keep your score sheet, but I need to get your answer sheet back. Tuesday and Wednesday I remember, but Monday I failed.
Oh, for your lab quiz, yeah.
Yeah, from the lab quiz.
If you want to just bring those to class tomorrow, that would be
Yeah,
fine.
that would be great.
Or if you have them now, you can leave them. So any questions? Any questions about DNA basic structure? The key components obviously are the fact that there are two strands of nucleotides that join together, that the DNA molecule consists of those strands wound around one another in a helical fashion, and that the two strands are joined together by what are weak individually hydrogen bonds, adenine and thymine, a pair of hydrogen bonds, guanine and cytosine, 3 hydrogen bonds. But collectively, those hydrogen bonds are strong enough as a group to give stability to this DNA molecule so it doesn't just fall apart. However, it really is significant that those bonds are individually weak hydrogen bonds, because at some point in time that DNA molecule does have to separate. And when it has to separate is during the process of replication. When Watson and Crick became convinced that their double helix model was indeed the correct model for the structure of DNA, they were pleased, because they were able to look at that model and say, hey, I think I know how this replicates. And the idea behind the replication was that the two parent strands separated from one another temporarily, and that each strand served as a copying surface or a template, and the sequence of nucleotides in each of those two parent strands dictated what the newly made strand was going to be, because of that rule of AT pairing with one another and GC pairing with one another. And so it really just made a lot of sense that DNA happened in this semi-conservative replication manner. And remember, semi-conservative refers to the fact that when we start out with one parent DNA molecule, which we can just represent as two solid lines, that replication then will give two daughter molecules which have conserved one of the strands of the DNA molecule of the parent, but the other being newly made. The same thing over here in this daughter right here. So this particular method of replication, they said, was a semi-conservative manner of replication. Well, I said something at the end of the period which applied to photosynthesis, and a lesson that people learned about photosynthesis. Just because something's logical does not necessarily mean it's true. So inquiring scientists said, yeah, this makes sense, but is this really indeed what is happening? Is there any experimental evidence that would support a semi-conservative model of DNA replication? Well, as people pondered DNA replication, they said, semi-conservative is one way in which DNA replication could occur. And it's a way that makes a lot of intuitive sense. But if we're going to be thorough, we should also consider the fact that there could be other methods of replication, and those other methods of replication were described as conservative replication and dispersive replication. So if we're going to be thorough, we really have to consider three possibilities. This one makes most sense, but we can't dismiss these out of hand without experimental evidence that says, sorry, this is not the way it's happening. Well, Messelson and Stahl conducted a very elegant experiment that evaluated these three models or hypotheses of DNA replication. So, here's what DNA replication would look like by these three models or hypotheses. We've already talked about semi-conservative. Parental DNA, one strand serving as a template for a newly made strand, other parent strand serving as a template for a newly made strand. So here's what DNA replication would look like in a semi-conservative fashion after DNA had replicated once. Okay? This is what DNA replication outcomes would be after this DNA had replicated again. So 1 round of replication, 2 rounds of replication. Let's walk through it, make sure you understand what's happening. So do you all see how you get from here to here? Does that make sense? Okay. So now what's happening over here? Okay, these 2 strands are separating, and this strand is still being used to make a newly made strand. So the light blue are the newly made strands. The dark blue are the original parental DNA. This light blue strand is now a template for a newly made strand. The same over here, the same over here. So after 2 rounds of DNA replication, we get 2 different kinds of DNA. Here we get a hybrid, 1 part old, 1 part new. Here we still get some of that hybrid. But we also then have a new kind of DNA. It's all new. Okay? No parental strands are found in either of these DNA molecules. Does that make sense, how that outcome is achieved as a result of semi-conservative replication? So this would be the prediction of what you would get if you let DNA replicate once, and this would be the prediction of what you would get if you let DNA replicate an additional time, 2 rounds of replication. Or if the DNA were bacterial DNA, first generation, second generation. Okay? Okay, what does conservative say? Well, conservative says that DNA replication is occurring by some mechanism whereby all of the original parental DNA ends up in one daughter molecule, and the other daughter molecule is all new DNA. Now, you might say to yourself, well, how could that happen? And it's hard to envision how that might happen. But you still have to account for that particular possibility. If we then let this DNA replicate again by the conservative model, we would get this particular outcome. Okay? So this parent would give this. So what you can see here is that after 1 round of replication, you would get a different outcome of types of DNA. And after 2 rounds of replication, you would get a different outcome of types of DNA as well. Okay? So these different models yield different predictions for outcomes. Okay, what does dispersive say? Well, dispersive says when DNA replicates, the 2 daughter molecules are a random mixture of some old and some new. And then when these replicate again, their offspring or daughter molecules would be a random mixture of some old and some new. So do you see how, particularly after the second round of replication, the predicted outcomes are quite different? Here, this is fairly similar to this, isn't it? A mixture of old and new. This is quite different. So that's what Messelson and Stahl relied upon. And they said, if indeed one of these 3 particular methods of replication is what is happening in living cells, then this is the outcome that we should get if we can let DNA replicate. And if there was some way to analyze what the DNA molecules that are produced by replication are made of. Are they made of old material, parental material, or are they made of new material? So how do they do that? Well, they also used isotopes in their research. And the isotopes they used were isotopes of nitrogen. This is the most common naturally occurrent isotope of nitrogen, nitrogen-14. There is a variant of nitrogen-14 called nitrogen-15, One difference in atomic number. Do these have the same or different number of protons? N14 and N15. Well, it's nitrogen, right? So they're going to have the same number of protons. So what's going to differ?
The neutrons.
Number of neutrons. Okay, so this has an extra neutrons. Do neutrons have mass? Yes, they do. They have an atomic mass of 1. Okay? So N15 would be heavy nitrogen, and then by designation, this is considered to be a light form of nitrogen. What's a technique we can use to separate things based upon differences in density? Centrifugation. Okay? So they use the techniques of centrifugation and then growing these bacteria. They studied DNA replication in bacteria. They did E. coli. And basically they were able to grow bacteria up in solutions of nucleotides. Do nucleotides have nitrogen in them? Yeah, they do. And the bacteria would incorporate these nucleotides into their DNA. And if you gave them N14 nucleotides, they would incorporate those. If you gave them N15 nucleotides, they would take those up and use them to make DNA. So this is the same thing I just showed you before, so we don't have to go through this again. So this is what we would predict after one round of replication and 2 rounds of replication. Average generation time for E. coli is about 20 minutes. So this would represent what would happen if we took a parental cell and then after 20 minutes, its DNA had replicated once, and then after 20 more minutes, its DNA would have replicated again. Here's what we would have expected with semi-conservative replication. A different outcome, a different predicted outcome. And then this would be dispersive. So, here's the experiment. They started out by culturing bacteria in media containing the N15 nitrogen nucleotides. So heavy nitrogen. Okay? And they let those cells grow in there. And so the original parent DNA would have been eventually that N15 form. And then they took those bacteria out and put them in a new environment. They put them in an environment where there was now N14 nucleotides. So what are they going to do? They're going to use those nucleotides to make their new DNA, the new daughter strands. So the question is, what would be the outcome? So they let them move into this environment where there's a new source of nitrogen, and then they let them sit for 20 minutes, isolated the DNA from the cells, and then separated the DNA by centrifugation. Okay, so these are the predictions that you would get for the 3 different types of replications after one round of exposure to these light nucleotides. This is what conservative would look like. This is what semi-conservative would look like. This is what dispersive would look like. So they did the experiment. And they were able to analyze the DNA and to see where it banded in the centrifuge tube. What would be the lightest DNA? The lightest DNA would be newly made DNA strands with the light nitrogen. The heaviest DNA would be the parental DNA with the heavy nitrogen. And bands in the middle would be hybrids of part light and part heavy, part old and part new. So they did the experiment, and what did they get? They got one band. So what did one band argue against? Conservative replication. Conservative replication said you should have 2 bands. One original parental and one brand new made. But this first experiment of 20 minutes exposure, one replication round, did not differentiate between the semi-conservative and the dispersive. because those both said you'll get a hybrid band. Okay? So what did they do? They did a second experiment, and instead of transferring this into the light nitrogen for 20 minutes, they did it for 40 minutes, 2 rounds of replication. After 2 rounds of replication, what would you predict for conservative? This. What would you predict for semi-conservative? This. What would you predict for dispersive? This. Here you would predict 1 light band, 1 heavy band. Here you would predict 1 light band, 1 intermediate band. Okay? Remember? So that's the first round of replication. Okay. So light band, this DNA, intermediate band, this DNA. What would dispersive predict? Still 1 band of DNA, a mixture. What did they get? They got 1 light band and 1 intermediate band. So that was the only outcome that was consistent with a particular model. And this provided direct experimental evidence that DNA replication was indeed happening by the semi-conservative fashion. So in this particular case, what was logical actually ended up being how things worked. But we had to verify that. We had to make sure that indeed that was really what was happening. So back to this slide I showed you before. It appears that DNA replication is pretty straightforward. And indeed what is happening is this double helix is separating, and this parent strand is serving as the template to make a newly synthesized strand, and that this parent strand is used as a template to make a newly synthesized strand. And what makes sure everything is properly paired is those base pairing rules must be satisfied. A has to pair with T, and G has to pair with C. Well, as it turned out, things weren't quite as straightforward as this would lead you to believe. And unfortunately, somewhat for you, DNA replication in nature doesn't really happen exactly like this. What's the biggest problem that we have right here in this DNA replication? There's 2 things that we have to consider. One is that the DNA molecule is anti-parallel. Remember what that means. One of the strands runs from head to tail, and the other strand runs from tail to head. Okay? So these 2 strands are anti-parallel to one another. The kicker is that DNA replication only occurs in the 5' to 3' direction on the new strand. So let's look at this strand over here to the left. This one's running in the 3' to 5' direction. So the newly synthesized strand is going to be this direction, right? So it can be replicated quite easily if these 2 parent strands separate in this 5' to 3' direction. Now these 2 molecules will be anti-parallel to one another. But can this strand be replicated in this direction as well? No, that's the wrong way. This particular strand is going to be replicated in this direction. So we're going to have replication going this direction on one strand, this direction on another strand. That's not what we'd envisioned. What we'd envisioned here is that these newly made strands are both going to be made in the same direction. You can't. That's a complication. That's a problem. Here's some other things we have to account for when we consider how DNA replicates in a living cell. One of the things we said that has to happen is that the DNA double helix has to separate. It has to unwind. Why does it only unwind during replication? Why doesn't it separate other times? Another thing we have to consider about DNA replication is it's really fast, particularly in bacteria. You can make a newly made strand at a rate of 500 nucleotides per second when you're assembling this newly synthesized strand. 1001. We've just joined together 500 nucleotides. That's cooking. It is slower in mammalian DNA, but still pretty fast, about 50 nucleotides per second. So how can this process be so fast? The other thing that's really important about DNA is that DNA replication is highly accurate. On the average, when you're making new DNA molecules, a mistake will only be made about 1 out of every 1 billion nucleotides that's added to these new molecules. That's pretty accurate, isn't it? Is it important that the replication be accurate? Absolutely. Okay, when you got your cells dividing, the DNA has to replicate prior to cell division, right? Both for mitosis and meiosis. Do you remember what part of the cell cycle that happens? We'll take you back to last semester. See Dr. Morton giving you looks here. Remember the cell cycle is broken up into the G, S, M phases. Where's DNA replication occur? Occurs in the S phase. That's where the chromosomes actually duplicate. So it's really important that if your cells are dividing, that the offspring have the same DNA as the parent cell did. We don't want errors in replication creeping in there. Nothing can be perfect, but a mistake rate of about 1 out of a billion is pretty good. The enzyme that actually catalyzes the addition of nucleotides into the new strand is the enzyme called DNA polymerase. So this enzyme is actually responsible for making a new DNA polymer. And this enzyme is kind of a funny enzyme. It's got some limitations. One of the limitations of DNA polymerase is that it only works in this one direction. It only adds new nucleotides in the 5' to 3' direction. So we're always adding to this 3' end as we extend the DNA. That's one limitation. The other is that to add nucleotides, it requires a 3' hydroxyl group. So on this 3' end, there's this hydroxyl group sitting out there. If that enzyme sees that hydroxyl group, it can stick another nucleotide onto there. But if it doesn't see a hydroxyl group, it can't. Well, when we're starting replication, this very first nucleotide, is there a hydroxyl group to attach that nucleotide onto? No, this was the first nucleotide put in there. So it can't start from nothing. There's got to be something there. That's what this de novo means, is that it can't start from nothing. There's got to be a nucleotide already there to add on to. Well, how do you do that when you're putting the first nucleotide into place? And then, because DNA is anti-parallel, the fact that this DNA polymerase only works in that one direction creates some problems. Because you can't just go like this up the DNA molecule as it unwinds. Because the enzyme won't work in opposite directions. So these are some things we have to consider. So let's go through now and see how these considerations or these requirements might be satisfied. Well, several of these considerations suggest that there are enzymes involved in DNA replication. And as it turns out, there are a number of enzymes and other proteins that must be present for DNA replication to occur. So, consideration one, DNA must unwind prior to replication. How's that going to happen? Well, it requires an enzyme. There is an enzyme called DNA helicase, also sometimes known as DNA unwindase, which is responsible for separating the two strands of DNA from one another so that those parent strands can be used as a template to make a newly synthesized strand. What's going to happen for those DNA strands to separate? You're going to have to break some hydrogen bonds, right? So this particular enzyme is going to catalyze breaking the hydrogen bonds. Now, this happens progressively. And so DNA starts unwinding, but as these hydrogen bonds down here are broken and the two strands separate, the ones farther up the molecule are actually still held together. That creates some interesting problems for DNA. Because as you start unwinding down here, if these parts are still held together, what happens to these parts that are still held together? They get all twisted up. So the part up here of DNA that's still hydrogen bonded starts getting all twisted and kinked as this part down here starts unwinding. That's not good for the DNA molecule, and in fact, that could probably interfere with further unwinding if this end gets too tangled up. So there's another enzyme called topoisomerase. And what that does is it temporarily nicks some bonds in this part of the DNA molecule and allows that stress of twisting up and stuff to be relieved until the DNA molecule starts unwinding to that point. So it relieves these kinks that develop as DNA starts unwinding. So that topoisomerase is really important. What normally holds those two strands together in the parent DNA molecule? Hydrogen bonding, right? Between A and T and G and C. So if we separate the two strands, won't they have a tendency to just come back together again and rebond? Yeah. But what happens as the DNA is unwound is there are some proteins that the cell produces which temporarily bind with the two strands and keep them from coming back together again. Those particular proteins are called single-stranded binding proteins. So 3 critical enzymes right there just to get the DNA unwound so replication can occur. Okay, we said replication was fast. When something happens pretty fast, oftentimes that is an indication that there's some enzymes that are catalyzing a process, increasing the rate of the reaction. The main replication enzyme that actually adds nucleotides to a growing strand is this enzyme DNA polymerase. And I can remember when DNA polymerase was discovered. Do you remember when it was discovered?
Not when it was discovered.
Yeah, there was a big article on the cover of Life magazine, and it basically said life in a test tube. And what they had done is they had isolated this DNA polymerase, and what they were able to do once they'd isolated this enzyme is take some parent DNA, denature it a little bit so it's separated into two strands, add some nucleotides to the test tube, and add this enzyme. And what this enzyme did was then copy the DNA. Well, that's not life, but it was a very remarkable accomplishment in, I think, late 1950s, mid 1950s. And this idea that we had an enzyme that could copy the stuff of life was really pretty remarkable. Well, as it turns out now, there are actually 11 different forms that we've identified so far of DNA polymerase. We'll talk about what some of these other forms of DNA polymerase do in just a minute. The major enzyme that's involved in DNA replication and actually hooking nucleotides together is a form of polymerase called DNA polymerase 3. Another thing that helps to speed up the rate of DNA replication is that it tends to be bidirectional. And it can occur in two directions at the same time. I'll show you why that's the case in a minute and how we can do that. So, here's what DNA replication looks like. Here's one of the two original parent strands, dark blue. This is a template. It's running in this 5' to 3' direction. So here's our newly made strand, light blue. Okay? These are new nucleotides that are being added. DNA polymerase is going to go in and say, this is a T, so I've got to bring in an A. Okay? This is a G, so I've got to bring in a C. C, G, A, T. And the enzyme brings in new nucleotides and then catalyzes the formation of this bond that joins the A to the C. Remember what the name of this bond is? This is the phosphodiester bond. Okay? And notice each time that the new nucleotide is being added to the 3' end, the hydroxyl group, of the previous nucleotide. So this T right here, its phosphate is going to attach to this 3' hydroxyl group. So replication of the new strand is occurring in the 5' to 3' direction. It's kind of interesting. This particular nucleotide is the triphosphate form of the nucleotide. A base, thymine base, a sugar, and a phosphate. So this is thymine triphosphate. What was this? This was a guanine triphosphate. What was this? This was a cytosine triphosphate. What was this? This was an adenosine triphosphate. Where have you heard adenosine triphosphate before? What's your shorthand for adenosine triphosphate? ATP. Okay, so we use ATP as the substrate for DNA synthesis. We use guanine or guanosine triphosphate as a substrate. The enzyme is going to chop off 2 of the 3 phosphate groups. And they're released in the form of this 2-phosphate group structure called pyrophosphate. And then the 3rd phosphate joins to the hydroxyl group of the previous nucleotide. I said that DNA could occur in a bidirectional fashion. What does that mean? What that means is if this is a strand of parental DNA, dark blue, and we're making newly made strands, light blue. A structure that we'll look at in a little bit called a replication fork develops. And then replication occurs in both directions away from the replication fork. Actually, several of these replication bubbles form on the DNA molecule at the same time. So a bubble forms here, and then we replicate in both directions away from the bubble. A bubble forms here, and we replicate in both directions. Another one here. And eventually, these bubbles all form together, merge together, and effectively, we have copied this DNA molecule much faster by having these multiple replication bubbles than if we just started at one end and move to the other end, step by step by step. So the formation of multiple replication forks and bidirectional replication helps to explain the speed at which DNA can be copied. This is an electron micrograph showing 3 replication bubbles that are forming as DNA is being copied. So it's not quite as straightforward as we originally thought. We originally thought the DNA just kind of unwound from one end, replication occurred, and we just kept unwinding and winding and winding until we got to the other end. Not quite that straightforward. What about the accuracy? Well, as it turns out, some of these other forms of DNA polymerase, remember I said that there were 11 of them, other than DNA polymerase 3, have proofreading capability. They are able to carry out something called mismatch repair. So what would we normally expect would happen as DNA was replicated? If there was an A here, there would be a T here. If there was a G here, there would be a C here. But what if this is the parent, and this is the new strand over here. What if an enzyme made a mistake and put an A in there instead? It could happen. It's not likely, but it could happen. And then it did the next one right, and the next one right. But this would be an error. Other forms of DNA polymerase can come along and check this molecule and go, yep, okay, not okay. And so what it'll do is it'll come back after the fact and remove the mistake and correct it. It's kind of like autocorrect when you're typing on your word processor. And sometimes you will hit the wrong key, but the software program will say they really meant the, and they will correct it for you and make that particular change. Okay. So some of these DNA polymerase molecules have this autocorrect capability, and they are able to carry out that mismatch repair. Okay, so limitations of DNA polymerase. It can't start from nothing. It can't start de novo. It needs a hydroxyl group to attach a nucleotide onto. That's a problem if you're the first nucleotide in a new strand. So what's the solution? Another enzyme. That enzyme is an enzyme called RNA primase. A little bit different here now. Remember, the main replication enzyme was a DNA polymerase. I mean, we're making new DNA here, right? But this enzyme is RNA primase. What does that suggest? That suggests that the first few nucleotides that are added into a new DNA molecule are RNA, not DNA. So if we talked about this, what's the difference between RNA and DNA? What does DNA stand for? Deoxyribose nucleic acid. What does RNA stand for? Ribonucleic acid. So there's a little subtle difference in the sugar of DNA and RNA. What's another big difference between RNA and DNA?
It uses urine.
We have a different nucleotide in place of thymine. We have uracil instead. So the 4 nucleotides are A, G, C, and U. And that's going to be important for exam questions. Anytime you see U, we are talking about RNA. Anytime we see T, we're talking about DNA. Okay? So DNA replication actually begins by adding a few nucleotides of RNA onto the template, or across from the template. Typically 5 to 10 of them. Now, does that mean that all of your DNA starts out with little bits of RNA? Well, initially it does. But eventually a repair enzyme is going to come back and cut out that primer and replace it with some DNA. So all replication of new strands begins with a short RNA primer. Why? Because RNA primase doesn't have that limitation. It can start from nothing. It doesn't need a 3' hydroxyl group to stick another nucleotide onto. When those primers are cut out and we stick new nucleotides together, how do we stitch together that little piece of new DNA that's been replaced with the part that's now being formed by DNA polymerase? We need an enzyme. We need another enzyme called DNA ligase. DNA ligase joins short pieces of DNA, or short pieces of DNA and long pieces of DNA, together. Ligate means to join. DNA ligase joins two pieces of DNA together. Really important enzyme we're going to see in a minute. So, here's our template strand running in the 5' to 3' direction. Here's our newly made strand running in the 5' to 3' direction. The RNA primase then will begin adding new nucleotides together and forming this short RNA primer in its 5' to 3' direction. Once the primer's there, DNA polymerase can take over and start adding DNA nucleotides now. So we just keep extending the length of this new strand as the polymerase moves down the template. Eventually, that primer will be cut out. Another form of DNA polymerase will fill back in these nucleotides, and then DNA ligase will join this little short strand of DNA to this other strand of DNA that's being formed. So several different enzymes involved in this process. But now we've got this big problem. The fact that the DNA polymerase is only working in the 5' to 3' direction. And the fact that DNA is anti-parallel. So in reality, when DNA is being replicated, one newly made strand is being made this way, and the other is being made this way. They're both going 5' to 3', but they're both going in opposite directions relative to the beginning of the original parent DNA molecule. We're going like this. That's not as nice and clean as going like this, because as you unwind the DNA, you can just keep replicating both strands in a continuous fashion. So what's going on here? Well, what would be nice is if we said, okay, maybe there's another form of DNA polymerase. Maybe there's a 5' to 3' form, and maybe there's a 3' to 5' form. And then we can just untwist DNA at one end, and both strands can go in the same direction relative to the end of the molecule. And people looked for years for a 3' to 5' DNA polymerase and have never found one. There appears to only be that form that works in that direction. So we're kind of stuck with having to do this. So what's that going to look like when we start copying DNA? Well, as it turns out, the two strands of DNA that are being copied, the two parent strands, are copied in a little bit different fashion. One of the two strands is called the leading strand, and it is copied in a continuous fashion, and I'll tell you what that means in a second. The other strand is a strand called the lagging strand, and this is copied in what we refer to as a discontinuous fashion. The lagging strand involves some of these short pieces of DNA being produced called Okazaki fragments. And they're little pieces of DNA about 200 nucleotides in length. If this were the leading strand, and this were the end of the DNA molecule, as the DNA unwound up here, this new leading strand could just continue to be produced, moving toward the replication fork until we reach the other end of the molecule. That would be the leading strand. That would be happening in a continuous fashion. On the other strand, the lagging strand, what do we have to do? We have to wait until the DNA is unwound a little bit, and then we back copy this direction. And then we have to wait till it unwinds a little bit more, and then back copy in this direction. And more, and back copy in that direction. That gives us these little fragments, these Okazaki fragments on that lagging strand. And the replication is discontinuous. There's kind of pauses while we wait for the DNA to unwind. Eventually, these Okazaki fragments are going to have to be stitched together. What enzyme is going to do that? The DNA ligase is going to do that. This one does start with the primer, but we only need one primer. And then we can just extend that strand continuously. Each one of these discontinuous fragments has to start with the primer. We're going to have a primer here and a piece of DNA. A primer here, a piece of DNA. A primer here, a piece of DNA. So we're going to have to cut those primers out and then stitch all those fragments together, again with DNA ligase. Well, we can show you, and this is probably a good place to stop next time, kind of a static version of how this looks. But what I'll